Good morning, students. Now coming to structure of TMV. You know, structure of tRNA. tRNA means actually RNAs of three types: mRNA, messenger RNA. Second one, rRNA, ribosomal RNA. Third one, tRNA, transport RNA. This is also called soluble RNA and adapter RNA. Okay. So from the beginning of genetic code, it was very clear to Watson and Crick. That is, from the most beginning of genetic code, it was very clear to Watson and Crick. Already, what is the contribution of these two scientists, Watson and Crick? Proposed double helix model for DNA. Okay, very beginning of the proposition of genetic code, it was very clear to this Watson and Crick that there is a special mechanism to read the genetic code and to link it to the amino acids. Okay, they discovered this thing, scientists Watson and Crick. From very beginning of the proposition of genetic code, it was very clear to Watson and Crick that there is a special mechanism to read the genetic code and to link it with amino acids. Why? Because there is no the special structural speciality for mRNA. There is no the special structures and special specialities to read the genetic code for amino acids and to read the genetic code for mRNA. So, there is a special mechanism to read the genetic code and to link it with the amino acids, because amino acids, they have no any structural specialities to read the genetic code. Okay. Similarly, this type of tRNA, transport RNA, it also functions as it performs dual functions. It performs dual functions. How it performs the dual functions? On one hand, this tRNA can read the genetic code. Very, very important. This tRNA can read the genetic code. Okay. On one hand, this tRNA can read the genetic code. On other hand, it can transfer the amino acids to the regions of ribosomes. So, tRNA performs dual functions. Not only to read the genetic code, what is the first codon and what is the next codon, what is the third codon, like that. It not only read the genetic code, but also it transfer the amino acids from cytosol to the region of ribosomes. That is during the time of protein synthesis. So, tRNA, it functions dual functions. tRNA, it functions dual purpose. At the same time, this type of tRNA, okay, this tRNA is also called the adapter molecule. This is also called adapter molecule. At the same time, this is also called soluble. It is a soluble RNA. This is a soluble RNA. And it accounts 10 to 15% of the total cellular RNA. 10 to 15% of total cellular RNA. Similarly, this tRNA is also represented by very limited number of nucleotides. What about the number of nucleotides? 75 to 90 nucleotides. They are present in 75 to 90 nucleotides are present in tRNA. So, this is also called adapter molecule. It's played very significant role in the transportation of amino acids from cytosol to the region of ribosomes during the protein synthesis on one hand. On the other hand, it can read the genetic code, what is the sequence of codons in mRNA. At the same time, this is also called adapter molecule, and this is soluble RNA, and this is represented by 10 to 15% of total cellular RNA. It is represented by 75 to 90 limited number of nucleotides. At the same time, this is also considered as the smallest RNA present in this is considered as smallest RNA. Okay, largest RNA rRNA. Then it is followed by mRNA. Least type of smallest type of RNA is tRNA. Okay, so to explain the structure of TMV, to explain the structure of this tRNA, scientist Coley proposed double two-dimensional secondary structure for tRNA. Scientist Coley proposed two-dimensional secondary structure. Okay, a two-dimensional secondary structure for tRNA was proposed by scientist Coley. Okay, this is a two-dimensional structure and a secondary structure, and this structure proposed by Coley, this is considered as a clover leaf model. Clover leaf model of tRNA. Clover leaf model of tRNA. So, clover leaf model of tRNA was proposed by scientist Coley. This is a two-dimensional secondary structure for tRNA. Okay, Coley proposed clover leaf model for tRNA. According to this clover leaf model, there is a single strand of nucleotides. Now this single strand of nucleotide, it is enfolded itself, leading to the formation of total 5 loops, 3 main loops, 1 accessory loop and 1 tail. So, according to this clover leaf model, there is a single strand of nucleotides. This single strand is enfolded upon itself, leading to the formation of 3 main loops in addition with 1 accessory loop and tail. For example, this is a tRNA clover leaf model of tRNA. Okay, this is a clover leaf model of tRNA. This is a 5' region. Okay, and this is a 3' region. This is enfolded, leading to the formation of 3 main loops. Okay, and 1 accessory loop, and another one is another 3rd loop. In this way, so this is a TMV main. This is a tRNA structure. Is represented by 1 strand of nucleotides, which is enfolded itself, leading to the formation of 5 loops. 1 is accessory loop and 3 main loops, and other one is tail. In this way, there are total 5 loops. Okay, the region of the tRNA 5' and 3' region of tRNA. This is tail. The tail portion, that is the 3' region, is also represented by 3 unpaired nitrogen bases at the 3' region, CCA. there are 3 unpaired nitrogen bases at the tail portion, that is at 3 front position. So that this region is called CCA end. This is called CCA end. This CCA end is responsible for the attachment of amino acid during the transportation. So that this is also called amino acid binding site. CCA end is responsible for the attachment of amino acid. So amino acid binding site or CCA end. The loop opposite to this CCA end means this loop. This is also represented by 3 unpaired nitrogen bases. This loop is called anticodon loop. This is called anticodon loop. Okay, this loop is called anticodon loop. The loop present opposite to CCA end or tail or amino acid binding site. That is this loop. This is also represented by 3 unpaired nitrogen bases, and this is called anticodon loop. Students, very important here. Okay. The unpaired nitrogen bases present in an anticodon loop of tRNA, they are complementary with the codons present in mRNA. Okay, this anticodon loop, unpaired nitrogen bases present at anticodon loop, they are complementary with the codons of mRNA. That such loop is called anticodon loop. Similarly, the right side loop, this is responsible for recognizing ribosome, recognizing ribosome, and this is also called T-pi-C loop. This is also called T-pi-C loop. T-pi-C loop. It is responsible for recognizing ribosome. Once amino acid is attached with the CCAN, now this amino acid is transported to the ribosome. Now this loop will recognize the site, ribosomal site, to transfer the amino acids. Similarly, now this left side loop, it is responsible for recognizing enzyme. This is also called DHU loop. This is called DHU loop. Left side loop is called DHU loop. And what is the importance of this loop? It recognizes the enzyme. But what is the importance of enzyme? Enzyme is responsible for the activation of amino acid. Just before the transfer of amino acids to the ribosome, through this tRNA, it must be activated by using an enzyme that is called enzyme amino acyl synthase enzyme. So, one loop is represented by anticodons, another loop is recognized ribosome, another loop is recognized enzyme, and this is also called DHU loop. Similarly, students, very important. Okay, that is according to the old concept. Okay, according to old concept, the number of tRNAs are equal to the number of types of amino acids. Okay, that is 20 types. There are total 20 types of amino acids. Okay, according to the old concepts, total 20 types of tRNAs are present in cytosol. Total number of tRNAs are equal to the number of the types of amino acids. That is 20 according to old concept. But according to the new concept, okay, according to the new concept, number of tRNAs are equal to the number of sensible codons of mRNA. There are total 64 codons in mRNA. 61 codons are sensible and are capable of recognizing amino acids. Remaining last 3, they are ending codons. So, new concept. According to the new concept, total 61 types of tRNAs are present in cytosol. Okay, each sensible codon of mRNA, it has a specific tRNA. So, according to the new concept, total tRNAs are equal to the number of sensible codons of mRNA, that is 61. Students, at the same time, okay, scientist Kim, okay, scientist Kim also proposed a three-dimensional structure for tRNA. Three-dimensional structure for tRNA. It resembles inverted L shape. Inverted L shape. According to the three-dimensional structure of tRNA proposed by Kim, it is inverted. It resembles with inverted L, upside down L. That is a three-dimensional structure proposed by Kim. Students, you know, for example, this is mRNA. Okay, 5' region and 3' region. This is mRNA, and this is first codon, starting codon. That is AUG, and this is another codon. For example, UAC. For example, this is mRNA, and 5' region and 3' region. This is one of the codon, first codon AUC, and this is also one of the codon in 64 codons, that is UAC. Okay. According to the new concept, every sensible codon present in mRNA is represented by a specific tRNA. Expect the nitrogen base is present in an anticodon of tRNA responsible for this first codon. Actually, the anticodons of tRNA complementary with the codons of mRNA. Expect the nitrogen base is present in anticodons of relevant tRNA responsible for AUG codon. For example, this is relevant tRNA responsible for AUG codon. Okay. This is tRNA. And this is anticodons of tRNA. For example, this tRNA is responsible tRNA for AUG codon. Expect the anticodons of tRNA. What nitrogen bases are present in anticodons of tRNA? Anticodons of tRNA, they are complementary with the codons of mRNA. So A is complementary with U, and U is complementary with A, and G is complementary with C. U, A, C, they are present in anticodons of this tRNA. Now, and which type of amino acid is recognized by this tRNA? AUG codon, it recognizes methionine. Okay, methionine amino acid is present at this tRNA. Now, hydrogen bonds, they are formed between these codons of mRNA and anticodons of tRNA. Methionine. Similarly, expect the nitrogen. For example, this is one codon, and this is the tRNA responsible for this codon. This is tRNA. Expect the nitrogen base is present in anticodons of this tRNA, and what is recognizing an amino acid? U is complementary with A is complementary with U, C is complementary with G, and hydrogen bonds are formed between these codons and anticodons. UAC, it recognizes tyrosine, amino acid tyrosine. In this way, the anticodons of tRNA always complementary with the codons of mRNA. Anticodons of tRNA, they are complementary with the codons of mRNA. Students, now coming to another point, very important here, that is, in case of prokaryotes, okay, the first tRNA that carries the methionine, that is called initiating tRNA. Initiating tRNA. This is called initiating tRNA. The first tRNA that carries the first amino acid methionine, that is called initiating tRNA. Okay, that is also called methionine tRNA. This is also called methionine tRNA because it carries the first amino acid methionine and is responsible for the transfer of the first amino acid of first codon. But in case this is in case of eukaryotes, okay. But in case of prokaryotes like bacteria, this methionine, it is also contain one formyl group. Okay, methionine is also contain one formyl group. A formyl group is attached with the methionine in case of prokaryotes. The tRNA that carries this formyl methionine, that is called formyl methionine tRNA. This is called formyl methionine tRNA. Okay, first tRNA is called initiating tRNA that carries methionine. But in case of prokaryotes, this methionine is also consists of one formyl group. So that this is called formyl methionine tRNA.